Good morning class, welcome back to microbiology. This is Silvana O'Coney and today's lecture, lecture 2, is about prokaryotic cell structure and function. Many prokaryotes have a variety of structures outside the cell wall that can function in protection, attachment to objects, and cell movement. We're going to discuss some of these components. So for example, the capsule, S layer, and slime layer. These layers of material lying outside the cell wall have different names depending on their characteristics. When a layer is well organised and not easily washed off, it's referred to as a capsule. Capsules are usually composed of polysaccharides. When the layer is unorganised and easily removed, then it's called a slime layer. When the layer consists of a network of polysaccharides which extends from the surface of the cell, it's referred to as the glycocalyx, and this is a term that can encompass both capsules and slime layers because they're usually composed also of polysaccharides. In this photo here you can see several bacteria adhering to animal intestinal tissue by means of their glycocalyx. Many bacteria and archaea have a structured external layer of proteins or glycoproteins called an S layer on their surface. In bacteria, the S layer is external to the cell wall, but in archaea, it may be the only cell wall structure existing outside the plasma membrane. Although these external layers are not required for growth and reproduction and survival of the bacteria, they do offer several advantages when these bacterial species grow in their normal habitats. So for example, they're thought to help pathogenic bacteria resist phagocytosis by animal host phagocytes. These phagocytes are cells that would normally engulf and destroy invading bacteria. Classic example is provided by the bacterium Streptococcus pneumoniae. When it lacks a capsule, it's easily destroyed and doesn't cause disease. Whereas the capsulated variety, when it's injected into mice, it quickly kills them. Capsules also contain a great deal of water and can protect against desiccation. They exclude viruses and most hydrophobic toxic materials such as detergents. The glycocalyx also aids in attachment to solid surfaces, including tissue surfaces in both plant hosts and animal hosts. The S layer is thought to protect the bacterial cell against iron and pH fluctuations, against osmotic stress, enzymes, or even against the predator bacterium called Delovibrio, which preys on other bacteria. The S layer also helps maintain the shape and the rigidity of some cells. It can promote cell adhesion to surfaces. And finally, the S layer seems to protect some bacterial pathogens against host defences, thereby contributing to their virulence. Many prokaryotes have short, fine hair-like appendages that are thinner than flagella, and they're called fimbriae. These fimbriae mediate attachment to surfaces, and some types of fimbriae are required for twitching motility or gliding motility that occurs in some bacterial species. Twitching motility is seen as short, intermittent, jerky motions that are normally seen on very moist surfaces. And this provides some evidence that the fimbriae actively retract in order for the bacterial cell to move. Sexpili are similar to Fimbriae, except they're longer, thicker, and less numerous, only about 1 to 10 per cell. And sexpili are required for a type of mating exhibited by some bacteria called conjugation. In this electron micrograph, we can see both the long flagella and the numerous shorter Fimbriae. This is the bacterium Proteus vulgaris at 39,000 times magnification. Many of the motile prokaryotes move by use of flagella, singular flagellum. These are thread-like locomotor appendages which extend outward from the plasma membrane and cell wall. They can actually rotate very rapidly, just like the propellers on a boat. For example, E. coli flagella rotate at about 270 revolutions per second. Vibrio, as another example, Vibrio flagella rotate about 1100 revolutions per second. And the direction of movement of the flagella determines the direction of bacterial movement. So in general, counterclockwise rotation of flagella causes forward motion, which is called a run. And in general, clockwise rotation disrupts the run and it causes the bacteria to tumble. This is called twiddle. The flagellum is actually a very effective swimming device. You can imagine for a bacterium, swimming is quite a task because the surrounding water seems as thick and viscous as molasses. So the bacterial cell is actually boring through the water with its corkscrew-shaped flagella, and if flagella activity ceases, the bacterial cell stops almost instantly. This figure illustrates the relationship of flagellar rotation to bacterial movement. In A and B, we can see a forward run and a tumble by a bacterium that has a single flagellum. In C and D, we can see the same forward run and tumble depending on the direction of rotation of the flagella, and this bacterium has several flagella. Another type of motility is shown by a group of bacteria called the spirochetes. Spirochetes are helical bacteria that travel through viscous substances such as mucus or mud, and they do this by flexing and spinning movements, which is mediated by a special axial filament composed of flagella. A very different type of motility called gliding motility is employed by many bacteria, including the photosynthetic cyanobacteria. Although there's no visible external structures associated with gliding motility, it actually enables movement along solid surfaces at rates up to 3 microns per second. Now, bacteria don't always move around aimlessly, but they're actually attracted to nutrients such as sugars and amino acids, and they're repelled by harmful substances such as waste products. This movement is known as chemotaxis. If you were to place bacteria in the centre of a dish of nutrient agar containing an attractant, the bacteria will exhaust the local supply and then move outward, following the attractant gradient that they've created. The result is an expanding ring of bacteria. When a repellent is placed in the nutrient agar dish, the bacteria will move away from the repellent, creating a clear zone around the disc. Concentrations of chemical attractants and chemical repellents are detected by chemoreceptors on the surfaces of the bacterial cells. In this image here, bottom right of screen, we can see an example of positive chemotaxis on the left, and this is exhibited by E. coli consuming the amino acid serine. To the upper right of that dish, a smaller white spot. This is what happens when you have motile bacteria but non-chemotactic bacteria, and they're non-chemotactic because they're mutant bacteria that have lost that ability to exhibit chemotaxis. A colony to the lower right side of this nutrient agar plate, and you can see it there just as a tiny white spot. These are non-motile bacteria. The chemotactic behaviour of bacteria has been studied using a tracking microscope. This is a microscope that has a moving stage that automatically keeps an individual bacterium in view. In the absence of a chemical gradient, E. coli and other bacteria that have been studied move around randomly. For a few seconds, the bacterium will travel in a straight or slightly curved line called a run. When a bacterium is running, the flagella are organised into a coordinated corkscrew-shaped bundle. Then the flagella fly apart and the bacterium stops and tumbles. And this tumble results in the random reorientation of the bacterium so that it's now facing in a different direction. Therefore, when it begins the next run, it usually goes in a different direction. And this is illustrated in A. In contrast, when the bacterium is exposed to an attractant, it tumbles less frequently, or it has longer runs when travelling towards the attractant. Even though the tumbles can still orient the bacterium away from the attractant, over time the bacterium gets closer and closer to the attractant. And the opposite response occurs with a repellent. A number of bacteria can form a special resistant dormant structure called an endospore. Endospores develop within vegetative bacterial cells of several genera, including Bacillus and Clostridium. These structures are extraordinarily resistant to environmental stresses such as heat, radiation, chemicals, and desiccation. Some endospores have remained viable for around 100,000 years, and because some of them are dangerous pathogens, endospores are of great practical importance in food, industrial and medical microbiology, because it's essential to be able to sterilise solutions and also solid objects. In nature, endospores aid in survival when moisture or nutrients are scarce. And endospores are so impermeable that special stains had to be developed in order to visualise them, as you can see in this image. Now, the position of the endospore inside the mother cell, or sporangium, frequently differs among different bacterial species. So this makes it quite valuable in terms of identification at the species level. Endospores may be centrally located, as you can see here at the top right-hand side of the screen. The endospore may be close to one end, and that's referred to as subterminal, or it may be definitely terminal. Sometimes an endospore is so large that it swells the sporangium, and you can see that labelled as swollen sporangium, and it gives it quite a characteristic tennis racket appearance. Now let's look at the structure of an endospore, which we can see in the image at the bottom left of screen. This is the structure of an endospore in the bacterium called Bacillus anthracis. This is the causative agent of the disease anthrax. The spore is usually surrounded by a thin covering called the exosporium. Beneath this lies the spore coat, and the spore coat is composed of several protein layers and may be fairly thick. It's quite impermeable to many toxic molecules, and it seems to be responsible for the spore's resistance to chemicals. The spore coat is also thought to contain enzymes that are involved in germination of the endospore. The next layer is to the inside of the spore coat is the cortex, and this may occupy as much as half the spore volume. It's made of a type of peptidoglycan that is less cross-linked than the peptidoglycan that you find in the bacterial cell wall. The spore cell wall or core wall is found inside the cortex, and it surrounds the protoplast or spore core. The core of the endospore has the normal cell structures such as ribosomes and the nucleoid, but it is metabolically inactive. So why is it that an endospore is so resistant? Well, it's still actually not known exactly why the endospore is resistant to heat and other lethal agents. Although as much as 15% of the spore's dry weight consists of an acid called dipicalinic acid, which is complexed with calcium ions, and this acid calcium complex is located in the core of the endospore. And it was long thought that this complex was directly involved in heat resistance, but more recently they found that mutants that are still heat resistant lack the dipicalinic acid. So it might be that calcium does aid in resistance to wet heat and oxidizing agents, and it might be that the calcium acid complex stabilizes the spore's nucleic acids. In addition, specialized small DNA binding proteins are found in the endospore. They cover the DNA in the core of the endospore, and they seem to protect the DNA from heat and radiation and so on. Dehydration of the protoplast appears to be quite important in heat resistance. The cortex layer might osmotically remove water from the protoplast, thereby protecting it from both heat and radiation damage. The spore coat also seems to protect against enzymes and certain chemicals. Finally, endospores contain some DNA repair enzymes. So once the spore actually germinates, DNA is repaired by the enzymes and the cell becomes active again. So in summary, you could say that the resistance of endospores to environmental stresses is probably a combination of several factors. Endospore formation, also called sporogenesis or sporulation, normally commences when growth of the bacterium ceases due to lack of nutrients. It's quite a complex process, and it may be divided into 7 stages. Let's now take a look at the 7 stages of endospore formation. So firstly, in stage 1, a filament of nuclear material forms. In stage 2, there's an inward folding of the cell membrane to enclose part of the DNA and produce what's called the 4-spore septum. The membrane continues to grow and engulf the immature endospore in a second membrane, This occurs in stage 3, engulfment of forespore. Next, the cortex layer is laid down in the space between the two membranes, and both calcium and dipicolinic acid are accumulated. This occurs in stage 4. Protein coats are then formed around the cortex in stage 5. This is called coat synthesis. Maturation of the endospore occurs in stage 6. And then finally in stage 7, we have completion of coat synthesis and lysis of the sporangium. And this occurs as a result of the action of lytic enzymes. These enzymes destroy the sporangium and this releases the endospore. Sporulation requires about 10 hours in the bacterium Bacillus megaterium. The transformation of dormant spores into active vegetative bacterial cells is almost as complex a process as endospore formation. It occurs in 3 stages: 1, activation; 2, germination; and 3, outgrowth. And often a spore will not germinate successfully, even in a nutrient-rich medium, unless it has been activated. This image shows Clostridium pectinivorum emerging from the spore during the germination process. So in the transformation of dormant endospores into active vegetative bacterial cells, the endospore starts with activation. This is a process that prepares spores for germination and usually results from treatments like heating. It's then followed by germination, which is breaking of the spore's dormant state. This process is characterized by swelling of the endospore, rupture or absorption of the spore coat, and loss of resistance to heat and other stresses. Also an increase in metabolic activity. Many normal metabolites or nutrients such as sugars and amino acids can trigger germination after activation. Germination is then followed by the 3rd stage called outgrowth, and this is where the spore protoplast makes new components. It emerges from the remains of the spore coat and it develops once again into an active bacterial cell. So that concludes this lecture. I'll see you next time.